Welcome to Coaching 485, Capstone in Strength and Conditioning. This first chapter and first lecture, as you can see there, covers a lot of ground. It covers the structure and function of body systems, and really what this first chapter in the Essentials of Strength and Conditioning, or Strength Training and Conditioning book consists of is the high points of PE Prof 201 and the high points of PE Prof 202. So it touches a lot or covers a lot of the musculoskeletal system, functions as a review of that. Also, we're going to cover or review the cardiovascular system and touch on very briefly the pulmonary system. So we've got a lot of ground to cover here. And again, a lot of this will be review, or actually all of this will be review from 201 or 202. So I will try not to tell you, or not to say remember 500 times, but there's a definite possibility that I will do that unintentionally. So I apologize in advance for that. All right. So with respect to the musculoskeletal system, so that system tells you, or the name of the system tells you the components, right? So musculoskeletal includes the muscles and the skeleton. So as you can see there, the musculoskeletal system consists of the bones, muscle, joints, and then tendons. Remember that tendons are the attachments of muscles to bones as opposed to ligaments, which connect bones to bones. So the tendons serve to attach muscles to bones. And then also the joints, or joints broadly speaking, are the intersection of two or more bones. It's where two or more bones interact. So as far as the skeleton itself goes, the skeleton has two large components to it, the first being the axial skeleton. The axial skeleton consists of the skull, the spine, the ribs, and the sternum. And so one of the things about the spine, so you'll hear the spine referred to in several different ways. I myself will refer to the spine in several different ways. So one of those ways that you'll hear it referred to, or you may see it referred to in this text as well, is the vertebral column. So remember, your vertebrae are your spine bones, and so the vertebral column, a column is something that runs straight up and down. So the vertebral column then is all the spine bones sitting atop each other. So the axial skeleton then, skull, spine, or vertebral column, ribs, and sternum. The appendicular skeleton, your appendages are your arms and your legs. So the appendicular skeleton is all of the bones that are associated with the arms and the legs. So in the upper body, the appendicular skeleton starts with the clavicle, which is your collarbone, and then the scapula, which is your shoulder blade, and then everything distal to that. So everything farther from the body than that. So it starts in the upper body with clavicle, scapula, humerus, and then everything distal to that. In the case of the lower body, the appendicular skeleton starts with the pelvis, so the right or left innominate bones, and then the femur, and then everything distal to that. So the appendicular skeleton deals with everything related to the arms and the legs, or all the bones related to the arms and the legs. Joints are the junctions or intersections between bones. Typically there's just two, but in some cases there are multiple. In terms of the structural classification of joints, so the way those are laid out there, from fibrous to cartilaginous to synovial joints, are in order from least movable or least mobile to the most mobile. So in the case of fibrous joints, you have a direct contact or direct connection between two bones. There is no joint space, and typically those bones are joined by really thick ligaments. So for example, if we think about the skull, if we think about bones like the frontal bones and the parietal bones, there's a little jagged line between the intersections of those bones, and so those jagged lines in the skull are called sutures. And so obviously, we don't want those bones that form the cranium, form the top part of our skull, we don't want those to be able to move. So there's a lot of congruity between, for example, the frontal bone and one of the parietal bones. And so they fit together kind of like puzzle pieces. And then in addition to the way that they fit together nice and closely, there are also some ligaments that help secure those bones together so that we don't get any movement. For the most part, we're not going to talk about fibrous joints in exercise science. Or strength and conditioning, we often don't really talk about fibrous joints because they don't move. And so because they don't move, they're not really something that we're trying to exercise. They're not something that's very involved in exercise. Cartilaginous joints, you've got two bones joined by cartilage. And so cartilaginous joints could be joints where the two bones or ends of a bone are joined by hyaline cartilage. So remember that hyaline cartilage is the most abundant cartilage in the body. A special type of hyaline cartilage is your articular cartilage that lines the ends of long bones inside of a joint. And in the case of growth plates, the proximal end of a bone and then the shaft of a bone are separated by a plate of hyaline cartilage, which is the growth plate or epiphyseal plate. So that's actually an example of a type of cartilaginous joint. After we've reached skeletal maturity, and in fact throughout the lifespan, we have cartilaginous joints in the vertebral column. So for example, between two of your vertebrae, two of your spine bones, you'll have the articular discs, not the articular discs, sorry, the intervertebral discs. And so those discs are comprised of fibrocartilage that separates the two bones. So that would be an example of a cartilaginous joint, the interaction between a superior vertebrae, the intervertebral disc, and then an inferior vertebrae. That's a cartilaginous joint. And then also, since I think I have my pointer up, I do, right here on the anterior aspect of the pelvis, you can see this little ring there of fibrocartilage. So that's the pubic symphysis. So that's another example of a cartilaginous joint. In the case of the vertebral column, you get quite a bit of movement out of those cartilaginous joints. In the case of the one there in the front or on the anterior aspect of the pelvis, the pubic symphysis, we don't get very much movement, which is good because we don't really want a whole lot of movement there. The vast majority of the time in strength and conditioning, we're going to be dealing with synovial joints because synovial joints are the most movable. And so synovial joints have these characteristics that you can see at the bottom of the slide. So there's a joint space, so there's an obvious separation between the bones. The two bones are connected to each other by a joint capsule. So here's joint over here and then capsule over here. The joint capsule itself has two layers. The outer layer is called the fibrous layer. And so that's really thick collagen tissue. So sometimes you've got ligaments that are part of that external or superficial layer of the capsule. So that's the fibrous layer. It does the structural connection between the bones. And then inside of or deep to the fibrous layer, you've got the synovial layer, which is a membrane that secretes synovial fluid. Synovial fluid is up here on line 1. And so synovial fluid is a lubricating fluid, nice and slick, helps the bones slide on each other. In addition to that, synovial fluid also helps nourish things inside of the joint that don't have direct blood supply. So for example, the articular cartilage, primarily that. And then some intra-articular ligaments can get some of their nutrition from synovial fluid as well. But its primary job as far as we're concerned is to lubricate to help the bones slide on each other. Then we've got articular cartilage, which is, again, lines the ends of our long bones. It's a special type of hyaline cartilage, and it's kind of like Teflon in that it's really, I was going to say that it has a kind of a pearly white appearance. It's fairly thick, so it helps absorb shock. But with respect to it being like Teflon, the two ends of the bones, it allows them to slide on each other more easily. So if you've got Teflon on Teflon, it's going to move, those two bone ends will slide on each other pretty easily. So that's the job of the articular cartilage, absorb impact and to be nice and slick. And then the ligaments here, so ligaments connect bones to bones. So you can have ligaments inside of the joint capsule, outside of the joint capsule, or that are part of the joint capsule. But what ligaments do is they connect bones to bones and they help to give the joint some structural integrity. Speaking of joints, so joints can be categorized by degrees of freedom. Remember, see, there it is. Back in 201, we talked about degrees of freedom, and we talked about the planes of the body. So 3 planes of the body are the sagittal plane, divides the body into left and right. The frontal plane divides the body into front and back. And then the transverse or horizontal plane divides the body into top and bottom. And so with respect to degrees of freedom, a joint that has 1 degree of freedom moves in 1 plane. So for example, the hinge joint of the elbow, the humeral-ulnar joint, only moves in the sagittal plane because we only get flexion extension at that joint. And so that's a joint that has 1 degree of freedom. It is uniaxial. And so that's the joint that's pictured for you there on the slide. A biaxial joint is a joint that moves in 2 planes. So the example I've got on here is the second metacarpophalangeal joint. So your MCP joints, those are your knuckles in digits 2 through 5. So your second MCP joint is right here. And so at that joint, we can get flexion extension, which is in the sagittal plane. And then we can also get AB adduction of the fingers, which is in the frontal plane. And so because it can move in both the sagittal plane and the frontal plane, it has 2 degrees of freedom, and it is then biaxial, bi meaning 2. And then multiaxial joints move in all 3 planes. So those are usually your ball and socket joints. So things like joints like your shoulder and your hip. So the glenohumeral joint is your shoulder, acetabulo-femoral joint is your hip. And so both of those joints can flex and extend in the sagittal plane. They can AB adduct in the frontal plane, and then they can rotate medially and laterally in the transverse or horizontal plane. So they can move in all 3 planes. They have 3 degrees of freedom and thus are multiaxial. All right, the muscles themselves. So when we talk about muscles, we talked about, or the 3 pieces of information I wanted you to know for every muscle in 201 was the muscle's origin, the muscle's insertion, and then the muscle's action. So where does it start, where does it stop, and what's it do? So where does it start and where does it stop? The origin and insertion. Those are also known as the proximal and distal attachments. So typically, a muscle's origin is the proximal attachment, and the muscle's insertion is the distal attachment. Not always, but in general, that's how it works. And then if we were talking about trunk musculature, typically the origin is superior and the insertion is inferior. As mentioned a minute ago, tendons connect muscles to bones. So on either end of the belly of a muscle, that connective tissue, the collagen that we're going to talk about here in a second, those layers merge to form the tendon, and then the tendon will attach the muscle to a bone. Tendons themselves insert into the periosteum, which is the outer layer of a bone. The connective fibers or the collagen fibers of the tendon actually run in with the collagen fibers of the periosteum and help anchor the muscle to the bone so that when the muscle contracts, it can pull on the bone and cause it to move, cause it to rotate around another bone. Muscle cells and muscle fibers are the same things. I use those terms interchangeably, so just something to be aware of. If a question asks you about a muscle cell or a muscle fiber, it's referring to the same structure. In terms of the organization of a skeletal muscle, so the smallest layer of organization is the muscle cell, aka a muscle fiber. And so the muscle cell is pictured for you here. So this is a single muscle cell right here, down near the bottom. And then it has its own layer of connective tissue. And so the layer of connective tissue around an individual muscle cell is called the endomysium. And so the endomysium is pictured here. So the endomysium is a layer of connective tissue around an individual muscle cell. Then, from the cell, we can bundle several cells together, and we get what's called a fascicle, or a fasciculus. And so the fascicle has its own additional layer of connective tissue, which is called the perimysium. So the perimysium wraps around a fascicle and separates it from other fascicles. And then the largest level of organization in a muscle is the muscle itself. And so the muscle itself includes all of the fascicles, and again, in each fascicle there is a group of muscle cells. The muscle itself, or the whole muscle, another way of saying that, has a layer of connective tissue that surrounds it, and that layer of connective tissue is called the epimysium. So connective tissue around the whole muscle, epimysium, connective tissue around a fascicle, perimysium, connective tissue around an individual muscle cell, endomysium. So from superficial to deep, epi, peri, endo, and then mysium refers to that specific connective tissue. The neuromuscular junction. So the neuromuscular junction tells you what it is, right? So neuromuscular, the nervous system, and then the muscle, and then where they meet. So the neuromuscular junction then is the intersection of the nervous system and the muscle. More specifically, it's the intersection of an alpha motor neuron and a muscle cell. So an alpha motor neuron is one that controls or innervates skeletal muscle cells. So one that causes the contraction of voluntary muscle fibers, which are skeletal muscle cells or muscle fibers. The neuromuscular junction is also known as the motor end plate. You'll hear that, or you'll see that terminology from time to time. Other things to know. So this definition, everybody likes to ask about this one. I imagine the NSCA will too on their exam. But a motor unit is a single alpha motor neuron and all of the muscle fibers that it innervates. So one thing to know about motor units, in addition to the actual definition, is that motor units vary in size, and they can vary in size pretty greatly. So remember, a small motor unit will be one alpha motor neuron, which is pictured for you there on the right. So this is our motor neuron. A small motor unit will be one alpha motor neuron and maybe 8 to 10 to 12 fibers, right? So we're going to see motor units that are that small in muscles that require a really fine level of motor control. So for example, the muscles that move your eyes will have motor units that are that small, because we want really small movements if we're, let's say, tracking something with our eyes, or watching a bird fly in front of us or something like that. We want really small movements to be able to track that nice and smoothly. In larger motor units, you may have an individual alpha motor neuron innervate several thousand fibers. So we see that in the muscles of the lower legs, so particularly gastrocnemius, the quads, the hamstrings example, those will have some pretty large motor units in them. And with large motor units, when that alpha motor neuron depolarizes, it's going to cause all of those muscle cells to at least twitch, so they're all going to contract. And so if you activate a couple thousand muscle fibers at once, you're going to get a pretty big contraction. And so we innervate those large motor units when we want really big, really powerful movements. So motor units vary in size. Another thing to know about motor units is that an individual alpha motor neuron, this guy, is attached to, in humans, muscle fibers that are all of the same type. So an alpha motor neuron is attached to all type 1 or slow twitch fibers. A separate motor neuron is attached to type 2As. A separate motor neuron is attached to type 2Xs. Remember, your type 2 fibers are your fast twitch fibers. So the motor neurons then are only hooked up to or only innervate one particular type of fiber, whether that's fast twitch or slow twitch fibers. And here's a couple different motor units. So here's my little purple alpha motor neuron. It's sitting in the spinal cord. Its axon leaves the ventral horn of the spinal cord, goes out the anterior part of our spine here, and then it's going to join this spinal nerve, and then it runs all the way out to these two muscle cells. So the purple motor unit then is only 2 muscle cells, so it's a small motor unit. As opposed to the red one, you can see this neuron goes out here and separates and attaches to 3 different muscle cells. And so the red motor unit is the larger of the two, because the red alpha motor neuron innervates 3 muscle fibers, purple only innervates 2. So the purple one's the small motor unit. In this example, the red one is the larger of the 2 motor units. Looking more closely at a muscle cell, some basic review of the components of a muscle cell. So the first one there is the sarcolemma. So the sarcolemma is the cell membrane of a muscle cell. And then we have the sarcoplasm. So the sarcoplasm is kind of pictured. The sarcoplasm is the cytoplasm of a muscle cell. So it's the fluid that is here between all these structures. That's the sarcoplasm. The sarcoplasmic reticulum is this yellow spider web here. The thing to know about the sarcoplasmic reticulum, it contains calcium. Then we have our T-tubules. And so the T-tubules on most diagrams are a different color, but on this diagram they are not. So the T-tubules are here. They run perpendicular or at a 90-degree angle to the sarcoplasmic reticulum. The T-tubule's job is to distribute the change in electrical charge when a muscle cell is stimulated. They help relay that electrical charge that's going to be moving along the sarcolemma inside of the cell and then across the sarcoplasmic reticulum. So the T-tubules help conduct or convey that depolarization, that change in charge of the muscle cell when it's activated or stimulated. Then we have our myofibrils, and so each of these little cylinders here is a myofibril. Myofibrils are composed of myofilaments. The myofilaments are the contractile proteins in a muscle cell, and the 2 contractile proteins are actin and myosin. Myosin will attach to actin, pull it toward the midline of the sarcomere, which we'll talk about here in a second. That causes the myofibrils to shorten, and then as myofibrils across the muscle cell shorten, the muscle cell on the whole shortens, and as that happens across a muscle, then the muscle on the whole shortens itself, and then that is going to pull the proximal and distal attachments closer together and cause shortening of the muscle and cause movement. Speaking of sarcomeres, so as you can see there, the sarcomere is the smallest contractile unit of a muscle cell. Sometimes it's referred to as the functional unit of a muscle cell. And so the sarcomere runs from Z-line to Z-line. And so in individual muscle cells, you're going to see this repeating pattern of light and dark in skeletal muscle and also in cardiac muscle. But right now we're talking about skeletal muscle. So you're going to see this repeating pattern of light and dark. The light area is where there is not overlap of the myofilaments. The dark area is where there is overlap of the myofilaments. So the individual, or the smallest contractile unit here, the sarcomere, so you've got our Z-line here, Z-line here. I'm going to drop down to the colored diagram because it makes things a little easier to see. So the blue stuff here is actin, the red is myosin. We're going to disregard these other proteins for now. So Z-line here, actin in blue, myosin in red. And then the other areas of a sarcomere then, you can see starting at the Z-line, so I'm actually going to start on the left and I'm going to move toward the right. So we move from the Z-line to the I-band. The I-band is where there is actin only. So I-band is actin only. The A-band is where there's overlap, so it runs from one end of the myosin to the other. So you got lots of overlap in most of that area. And then the H zone is this middle portion with myosin only. And then the M line is the midline here. And those are the major structures of the sarcomere. So you got Z-line, which serves as the anchoring point for the actin. I-band, actin only. A-band, overlap of actin and myosin. H-zone, myosin only, and then the M line is the midline. And so you can see that up here on this fully relaxed sarcomere. So you've got our Z-line here. All this stuff, these lines, are actin filaments. And then we add in the myosin here. So we've got Z-line, I-band, A-band, where the two overlap. And then you can see the H-zone here in the middle, where there's only myosin, and then you can also see the M line right there where it's slightly darker. And then everything just repeats in a mirror on the other side. And so if we are able to zoom out, I don't have that drawing on this particular, I don't have that diagram on this particular slideshow, but if we zoom out a little bit, we would see that right here on the other side of this sarcomere, you're just going to get a repeat of that. And then that continues on the way down, or all the way down until we reach the end of the muscle cell. All right, speaking of muscle contraction, so the primary explanatory mechanism for muscle contraction is called sliding filament theory. And sliding filament theory, in short, says that myosin attaches to actin when the muscle cell is stimulated, pulls actin toward the midline. That causes the Z-lines to move closer together, which thus shortens the sarcomere. And again, as sarcomeres throughout the muscle cell shorten, the muscle cell in the whole shortens, and then we can extrapolate that up into the entire muscle. So we'll start up here. So again, the blue stuff is actin. The red here is myosin. So this is a myosin head. You'll see on the actin that we have these little indentations here, which are the binding sites that myosin really likes. And so there's two other proteins here. There is troponin and tropomyosin. So troponin is this yellow, or are these little yellow dots. That's your troponin. Again, I'll refer to it as troponin C, C for calcium. And then tropomyosin is this orange thread here. And so at rest, what's going to happen is that tropomyosin is covering up these binding sites on actin. But when the muscle cell is stimulated, the sarcoplasmic reticulum is going to release calcium. Calcium is going to bind to troponin C. Troponin C, because it's connected to tropomyosin, when troponin C changes shape after binding to calcium, it's going to pull tropomyosin off of these binding sites on actin. Once those are exposed, once myosin can see those binding sites, it's going to jump up and attach. And that attachment between actin, or starting here, between myosin and actin is called a cross bridge. So now we have cross bridge formation. And so once we've got cross bridge formation, myosin at rest is kind of like a coiled spring. So as soon as it sees those binding sites on actin, it's going to jump up and attach and then pull actin toward the midline. And that's called the power stroke. And so that's what you see happening here in this second picture. And then in order to get myosin to detach from actin, we have to add an ATP, which is adenosine triphosphate, the energy molecule. And so that's what you see happening down here. We've added an ATP. That causes myosin to detach from actin. And then what we're going to do is we're going to use an enzyme called myosin ATPase. ACEs are enzymes. And we're going to split that ATP. We're actually going to split off the last phosphate group. When we do that, that gives off free energy. The products, in addition to free energy that gives us and hydrogen, are adenosine diphosphate, or ADP, and then PI, which is a free phosphate. And so when we split that ATP, when we give off that free energy, that helps us reset the myosin head. That helps us sort of reset the spring, if you will. So myosin is in its position here, ready to spring again if it continues to be or continues to see those binding sites on actin that it really likes. So at rest, running through these, at rest, tropomyosin is covering up binding sites on actin that myosin really likes and myosin will attach to when it sees them. In terms of excitation contraction, when the muscle cell changes charge, so when the inside of it becomes temporarily negative, that's going to cause the release of calcium from the sarcoplasmic reticulum. Calcium is going to bind to troponin C, causes it to change shape. Because troponin is attached to tropomyosin, that'll pull the tropomyosin off of the binding sites on actin, exposing them. When myosin sees those, it's going to jump up and attach, pull actin toward the midline, and again, that pulling of actin toward the midline is called the power stroke. Then we're going to attach an ATP to the myosin head, get it off of, or get it to detach from actin, and then reset. Again, we're going to break down ATP, give off free energy, and then that'll allow us to reset the myosin head. And then, if we don't want the muscle cell to contract anymore, we'll stop getting signals from the central nervous system. The alpha motor neuron will stop depolarizing, so it'll stop sending electrical signals itself. And then when that happens, that is going to cause the pumping of calcium back into the sarcoplasmic reticulum, which then removes it from troponin C, allowing tropomyosin to cover those binding sites back up, so myosin has nothing to grab onto. And so that's what happens when the muscle cell relaxes, is that tropomyosin covers up those binding sites on actin, so myosin can't grab it and can't cause the muscle cell to shorten. All right, not doing too bad on time here. I'm talking kind of fast, although I always do that, but I'm talking kind of fast, and I'm trying not to make this into a 3-hour lecture, because again, it is all review from stuff you've had both in Structure 1 and 2, but also to some extent you've probably covered this in classes like 471 as well. All right, so neuromuscular system. So as discussed a minute ago, muscle fibers are innervated by motor neurons, specifically by alpha motor neurons, that transmit impulses, so electrical signals from the spinal cord. So in terms of activation of muscles, so now we're moving up from what happens inside of the muscle cell, and we're looking at the interaction of the motor neuron and then the muscle cell. So the first thing that happens is an action potential. So our alpha motor neuron, I don't have a picture, I thought I did, sorry. First thing that happens is that our alpha motor neuron is going to depolarize. So it is going to go from being negative to being positive very temporarily. So at rest, both the alpha motor neuron and the muscle cell are negative. And then when they depolarize, when they change charge, when they send an electrical signal, however you want to phrase it, they go from being negative to being positive very temporarily. I think a minute ago I might have made a mistake when I was talking about muscle cells, but this part is definitely correct. Muscle cells at rest are negative, and then they very temporarily go positive, and then at rest they go back to being negative again. So the alpha motor neuron is going to depolarize. That change in charge is going to go from the cell body of the alpha motor neuron down its axon out to its dendrites, eventually reaching the axon terminals. When that change in charge reaches the end of the axon terminals, That change in charge is going to allow an influx of calcium. You probably don't have to know this for the NSCA, but it'll allow an influx of calcium into the terminal. That causes vesicles, little basically bubbles, inside of, not bubbles isn't the best way to put it. They're phospholipid bilayers, right? And so they contain acetylcholine, which is abbreviated there ACH, which is a neurotransmitter. And so when you get that change in charge of the alpha motor neuron at the axon terminals, you get an influx of calcium into them, causes the release of acetylcholine. Acetylcholine is dumped into the synaptic cleft. And the synaptic cleft is the space between the end of the alpha motor neuron and then the muscle cell itself, or the sarcolemma of the muscle cell. When acetylcholine is dumped into the synaptic cleft, it moves across the cleft, attaches to what are called chemically gated channels. So chemically gated channels are channels that open up in response to receiving a chemical. So the chemical is the key. And so when acetylcholine binds with those chemically gated channels, those channels open up. They allow sodium, a positive ion, to move from the outside of the cell, muscle cell in this case, to the inside of the muscle cell. That causes a change in charge in that area. Once we get enough calcium, or sorry, I've switched your ions. Once we get enough sodium coming inside of the muscle cell, that is going to raise the charge of the sarcolemma enough that we're going to get the opening of these voltage-gated channels. And when the voltage-gated channels open, and they open in response to change in charge, so from when the muscle cell goes from being negative 90 to more like negative 55, when that happens, the action potential or the depolarization becomes self-propagating. So voltage-gated channels in one area open, allows a bunch of sodium to come inside. The neighboring voltage-gated channels then open, causing more sodium to come inside. And so it happens in a wave-like fashion like that, moving out from that motor end plate, that intersection of the alpha motor neuron and the muscle cell. So once we've reached threshold voltage, the voltage-gated channels open, and again, are going to allow that depolarization to become self-propagating. Then as the depolarization, the wave of change in charge spreads along the muscle cell, it's going to go down through the T tubules, across the sarcoplasmic reticulum, which is what SR is there. That change in charge causes the release of calcium from the sarcoplasmic reticulum. Calcium binds to troponin C. Troponin C pulls tropomyosin off the binding sites on actin, and then we get cross-bridge formation. So the latter half of that I talked about on the previous slide. And here's a depiction of all those things I just talked about. So this is the end of our alpha motor neuron. Here's the axon terminals. Here's our little vesicles that contain acetylcholine. These little green dots are acetylcholine. So again, change in charge allows calcium influx. That causes the vesicles to move here to the end of the axon terminals. They're going to dump their acetylcholine. It's going to bind to these chemically gated channels. Sodium comes rushing inside. And not pictured here, but you would have voltage-gated channels adjacent to these. And so again, once we hit that threshold voltage, which is somewhere around negative 55 millivolts, the voltage-gated channels open, causing neighboring voltage-gated channels to open. And so then you get this wave of change in charge where the inside of the cell goes from being negative to being positive very temporarily. All right. Motor units. So we talked about this stuff. One important thing to know about motor units that I hinted at but didn't say specifically is the idea of the all or none principle. So when an alpha motor neuron depolarizes and releases its acetylcholine, it's going to release that acetylcholine to all of the muscle cells that it interacts with. And so if an alpha motor neuron interacts with 2,000 muscle fibers or muscle cells, it's going to release acetylcholine to all of them, and all of them are going to at least twitch, which is to contract very briefly. So that's the all or none principle, is if I stimulate an alpha motor neuron, all the muscle fibers that it's attached to are going to be activated. They're going to contract. And so because of that, we recruit motor units from smallest to largest whenever I am lifting objects or whenever I am moving. So if I'm doing something that requires very minimal effort, let's say I'm lifting something fairly light, like a TV remote control or something like that, or my phone, those weigh only a few ounces. And so because of that, it doesn't require very much force from the muscles to be able to lift them up. So as a consequence, I'm only going to use the type 1 or the slow twitch, the smallest motor units. And then if I'm lifting something heavier, so rather than a phone, let's say I'm lifting a brick, something slightly heavier, so then I'll need additional motor units. So I'll need additional muscle fibers, so I have to recruit additional motor units, and so I activate additional alpha motor neurons. So that's motor unit recruitment. That we work on the size principle. We start with our smallest motor units. So to use this diagram here, we're going to start with the purple alpha motor neuron first. We'll get it to recruit its 2 muscle fibers. If that isn't enough to lift whatever it is I need to lift, then we will also recruit the second motor unit, the red one, that has 3 fibers, and so then we'll engage a total of 5 fibers in order to lift whatever it is I need to lift. Rate coding is the number of times per second the alpha motor neuron depolarizes. So it is the frequency of action potentials, or the frequency of depolarization, but basically what happens is if I need to either move really fast or produce a really strong contraction, what I'm going to do is to send lots and lots of electrical signals, again, aka action potentials or depolarizations, down this alpha motor neuron out to the muscle fibers. If electrical signals continue to pulse through the alpha motor neuron, it's going to continue to release acetylcholine. If it continues to release acetylcholine, that will continue to attach or bind to the chemically gated channels on the muscle cell, which will continue to allow sodium inside, which keeps the inside of the muscle cell fairly positive, which causes basically a continued release of calcium from the sarcoplasmic reticulum, which causes basically a continued binding of calcium to troponin C, which keeps tropomyosin off of those binding sites, which keeps the binding sites on actin exposed, and so you get this constant interaction of myosin and actin, because as long as myosin sees those binding sites exposed, it's going to continue to jump up and attach to those and pull them to the midline. So the more frequent those action potentials coming down the alpha motor neuron, the more consistent its release of acetylcholine, the more consistent the binding of acetylcholine, the more the inside of the muscle cell stays positive, and the more then calcium stays out of the sarcoplasmic reticulum, allowing the exposure of those binding sites on actin. So the way that increasing the force of muscle contraction works then is we start with our small motor units, our type 1 motor units first. Then we work our way up to the middle ones, the type 2As, and then eventually up to the type 2Xs. And then if we still need to produce more muscle force, we're going to employ rate coding. So we're going to increase the number of action potentials per second coming down the alpha motor neuron. So the 2 primary ways that we increase muscle force then, or increase our expression of strength voluntarily, is by increasing motor unit recruitment, recruiting larger motor units, and then increasing the frequency of depolarization of the alpha motor neuron, which is referred to as rate coding. Speaking of muscle fibers, so again, 3 types of muscle fiber, 2 broad categories of muscle fibers. We're going to talk about 1 subcategory. There are others, but we're only going to talk about 1. So in terms of the broad categories of muscle fibers, you've got slow twitch and fast twitch. And so what that refers to is the amount of time it takes for that muscle cell to reach peak tension. And so what you've got over here, all the way on the right, this is our slow twitch fiber. And so you can see that when it's stimulated, this is it developing tension, where my mouse is. So it takes about 100 milliseconds in order for it to reach peak tension. And when it does, it produces about 5 grams of force. So 100 milliseconds to produce 5 grams of force, as opposed to, over here on the left, this is our type 2X muscle cell. You can see when it's stimulated, it produces a lot of force, 50 grams of force, so 10 times as much, and it does it in less than 50 milliseconds, so in less than half the time. So our 2Xs then produce 10 times as much force, in less than half the time as our type 1. So the type 2Xs then, if we recruit those first, you can see why that'd be a problem. That's going to produce lots of force really quickly. So the type 2As then are kind of a middle ground. So you can see they produce 20 grams of force, or in this example, produces 20 grams of force in a little over 50 milliseconds, right? So a little bit slower than the 2Xs, but still quite a bit faster than the type 1 fibers. So the type 1 fibers then, our slow twitch fibers, are not very strong, so they don't produce much force, and they don't produce it very quickly. But an advantage that they have is that they are very fatigue resistant, so they can produce a little bit of force for a really long time. By really long time, I mean hours on end. So you use your type 1 fibers, for example, if you're going to run an ultra marathon. So if you're going to go run 50 miles, that's your type 1 fibers. So that's where we use type 1. And we also use them for most of our activities of daily living. So a lot of the stuff that we do doesn't require very much force. Again, picking up a phone or TV remote or opening a door, that kind of stuff. That doesn't require very much force, and so we can get by with our type 1 fibers, our small motor units, for most of the stuff that we do. Type 1 fibers, slow twitch fibers, are aerobic, so they're good at using oxygen to produce energy. The good part about that is there aren't a lot of byproducts of that, so it's not going to produce a lot of lactate or lactic acid. They're good at shuttling lactate or lactic acid out of the cell. What else? Oh, and then their fuels are pretty plentiful, right? So because they're aerobic, they can use fat for fuel, and our fat supply, even if you're fairly lean, is almost unlimited. Because fat is a really efficient fuel source. So they can fuel themselves, fuel that contraction for a long time, but again, low force development. On the other end of things, our type 2Xs, they're anaerobic, so they produce their energy using ATP and phosphocreatine, and then the glycolytic system, so they're going to break down sugar, not in the presence of oxygen. And so that's going to generate lactate or lactic acid. So they are anaerobic. They generate a lot of force really quickly, but because they're anaerobic, they fatigue really fast. So they're going to run through their ATP stored molecules pretty quickly. They're not going to deplete them, but they're going to run through them and decrease their supply of them fairly quickly, and then they will deplete their phosphocreatine stores within, give or take, 10 seconds. And so basically they run out of fuel. And so because of that, they fatigue really fast. They're going to then, once they've used up their phosphocreatine, they'll switch to the glycolytic system, but you can't produce as much energy as quickly using the glycolytic system. And then again, it produces lactate or lactic acid as a byproduct, and so the muscle cell fatigues fairly fast. Type 2As are the middle ground. And so they are, again, they don't produce as much force as the 2Xs, but they produce more than the type 1s. They have some mitochondria, so they are somewhat aerobic, but they also can operate anaerobically. So again, they're the middle ground in terms of force production, speed of force production, but also metabolically. And there's a helpful table. It's table 1.1 in the textbook, but I included it because I thought it would be helpful. So if you want to look at all the different characteristics of type 1 versus 2A versus 2X fibers, lots of information there. Proprioception. So proprioception is position sense. It's being aware of where you are in space. The proprioceptors that are important for the NSCA, for the CSCS exam, are muscle spindles and Golgi tendon organs. So muscle spindles are specialized muscle fibers that are inside of a muscle. So they are referred to as intrafusal fibers. They're special fibers inside of a muscle cell. Extrafusal fibers are just your normal fibers. And so with muscle spindles, so here's our muscle spindles here. So there are a couple different types of sensory endings attached to our muscle spindles. So you can see here our annulospiral endings, or the blue ones. And so the annulospiral endings wrap around the middle of the muscle spindles, and the middle of the muscle spindles kind of bulge out. Sometimes they're referred to as bags. And so the annulospiral endings then, what muscle spindles do is they detect muscle length and changes in muscle length. And on top of detecting changes in muscle length, they detect rate of change. So if the muscle is lengthening, how fast? Muscle spindles detect that. And so these annulospiral endings then that are wrapped around the middle portion of the muscle spindles, those detect rate of change in muscle length. So that's kind of their primary job, is detecting rate of change. So is the muscle being stretched? How fast? That's what they determine. As opposed to these little flower spray endings here, you can see they are the purple ones. So the flower spray endings there are on the ends of the muscle spindles, and they basically detect static length. So is the muscle lengthened or shortened? How much? That's what they detect. So basically the annular spiral endings are, kind of a way to frame this is, the annular spiral endings are while we're moving, flower spray endings largely are once we've stopped. They give us information about that. The muscle spindles are excitatory, meaning that if the muscles stretch too quickly, they're going to cause a reflexive contraction. So that's called the stretch reflex. So if we stretch a muscle really quickly, it's going to reflexively contract. You see that when you go to the doctor, if you're getting a physical, for example, and they do a reflex test. Most common reflex test is on the patellar tendon, so just below your kneecap. If they were to tap on that with the little reflex hammer, what that does is it causes a really quick stretch in the quadriceps muscle group. And then if everything is functioning normally, then that quick stretch is detected by the muscle spindles. The muscle spindles then will send an excitatory signal back to the spinal cord and cause a reflex muscle contraction. You can also do those reflex tests like on the triceps in the upper body and the biceps, and then brachioradialis is another common one in the upper body. So the thing to know about muscle spindles then, they detect muscle length, rate of change of muscle length. If a muscle is lengthened too fast, they cause reflexive contraction. Golgi tendon organs, on the other hand, and the tendon organ is pictured down here in yellow. As you would expect, they're part of the tendon, but they do the opposite. So at least with respect to muscle contraction. So Golgi tendon organs detect the amount of tension in a muscle, and if the amount of tension in a muscle gets too high, they cause reflexive relaxations. They cause the muscle to stop contracting. The idea is to keep the muscle from generating so much tension that it becomes damaged. So maybe, if you cause too strong of a muscle contraction, maybe you'll get damage to the muscle belly, but also maybe the tendon pulls away from the bone or something like that. So the Golgi tendon organs are kind of the governor on this whole system to try to prevent too much contraction of the muscle and try to prevent damage that way. The bottom thing there where it says overridden by PMC, PMC is primary motor cortex, it's in the brain. So one of the early adaptations, so since this is strength and conditioning, one of the things you'll see if you take somebody that is untrained or who is untrained and you have them start lifting weights, obviously they're going to be fairly weak when they first start, but you're going to see pretty rapid increases in strength, and you're going to see pretty rapid increases in strength within a couple weeks. But it takes several weeks in order for detectable hypertrophy or muscle growth to take place. So how are they stronger if their muscles aren't bigger? And the answer to that question, well, there are several answers to the question, but the one that's of relevance right now is basically they stopped hitting the brakes, the Golgi tendon organs, and their interaction with the brain have changed such that basically the brain is able to override that inhibitory signal, and so the muscle is able to contract more strongly before we hit the brakes. So basically with training, they stop hitting the brakes as early on, and they're able to generate more force, more tension, and thus lift more weight. All right, so that's the end of the musculoskeletal system. So on to the cardiovascular system. Hopefully you'll recall that the heart has 4 chambers. There are 2 atria and 2 ventricles, and then there are valves between the chambers to prevent backflow. Remember that when we talk about right and left in the heart, we're talking about the right side of the athlete or the left side of the athlete. So in this depiction, this is the right side of the heart. I know it's on the left side of your screen, but this is the right side of the heart because it is as if the athlete is looking at you. That would be the right side of their heart, and then this would be the left side of their heart. So here is our right atrium. Here's the left atrium. So the atria are the smaller of the chambers, and then here's our right ventricle. Here's our left ventricle. So the ventricles are the larger of the chambers. The valves, again, are going to prevent backflow. So here's one valve here, and then here's another valve here. So these two are called, so this one and this one, are called the atrioventricular valves, the AV valves, because they're between the atria and the ventricles. They prevent backflow from the ventricles back into the atrium. So there are 2 of those. There's a tricuspid and the bicuspid valve. Tricuspid valve is here because it's called tricuspid because it has 3 flaps. Bicuspid's over here. has two flaps. So the tricuspid valve is on the right side of the heart. The bicuspid or mitral valve is on the left side of the heart. Again, the way to remember that, or the way that I remember that, tricuspid is on the right side, so try to be right. And then mitral valve or bicuspid valve is on the left by process of elimination. Then we have our semilunar valves. So the semilunar valves separate the ventricles from the blood vessels that they empty into. So here you can see we've got our pulmonary artery. And so the semilunar valve between the right ventricle and the pulmonary artery is the pulmonary semilunar valve. And then our other semilunar valve is here, and so that's between the left ventricle and the aorta. And so that is the aortic semilunar valve. And again, those are going to prevent backflow back into the ventricles. In terms of path of blood flow through the heart, so remember that the right side of the heart receives deoxygenated blood from the body. It's not completely without oxygen, but it is certainly reduced in oxygen content. The right side of the heart receives deoxygenated blood. That's going to dump into the right atrium. Right atrium is going to contract, push the blood down to the right ventricle. Right ventricle is going to contract, push the blood out here into the pulmonary artery, takes the blood out to the right and left lungs respectively. We're going to drop off carbon dioxide of the lungs in the alveoli in particular. We're going to pick up oxygen. That freshly oxygenated blood returns back to the heart, specifically to the left atrium via the pulmonary veins. So then the pulmonary veins dump into the left atrium. Left atrium contracts, pushes blood down to the left ventricle. Left ventricle contracts, pushes blood out into the aorta. And then the aorta distributes blood to the rest of the body. So that's the path of blood flow through the heart. In terms of the electrical conduction system of the heart, so the SA node, the sinoatrial node, is also known as the pacemaker of the heart. It sits in the right atrium. It depolarizes on its own, and left to its own devices, i.e. without input from the central nervous system, the SA node would depolarize 100 times per minute. But of course, we don't have, or for most of us, our resting heart rate isn't 100 beats per minute, and so something else must be happening. And what's happening is that the SA node operates under the influence of the central nervous system, and so unless you're exercising, you're going to slow the SA node down a little bit so that your resting heart rate is somewhere around 70 or maybe 60 or maybe even lower if you're a well-conditioned athlete. So the SA node then is the pacemaker of the heart. It initiates the electrical signal in the heart. That electrical signal, again, that change in charge, that wave of negativity, or sorry, wave of positivity, didn't mean to mess you up there. That wave of positivity spreads across both of the atria. So it's going to spread across the right atrium here. You can see the connection to the left atrium over here. Causes the atria to depolarize, and then that electrical signal is going to pause very briefly at the AV node. So the AV node is going to pause that electrical signal for about 1/10 of a second, sometimes a little bit less, but give or take 1/10 of a second. And so the reason that's important is that allows the atria to fully contract, to push blood down to the ventricles before the ventricles contract. So that without that delay, the atria and the ventricles would basically contract at the same time, and obviously that would be problematic. So the AV node acts to pause the electrical signal, and then once we've done that, then the electrical signal, the depolarization, spreads really quickly across the AV bundle, which then spreads into or splits into right and left branches, and then branches out even farther into smaller fibers called the Purkinje fibers. And so what that does is we move into the bundle branches and then into the Purkinje fibers, the rate of depolarization actually picks up a little bit, moves a little bit faster, so that we stimulate the ventricles from the bottom up to wring blood out of the ventricles and push it out into the aorta or out into the pulmonary artery in the case of the right ventricle. As mentioned, the SA node is not left to its own devices unless you are a cardiac transplant patient. But if you don't meet that criteria, the SA node is under the influence of the central nervous system, specifically of the autonomic nervous system. The autonomic nervous system controls all the automatic motor functions in the body, so autonomic, automatic, and again, related to motor function. There are two branches to the autonomic nervous system. There's the sympathetic branch, which we associate with fight or flight. In the case of this diagram, the sympathetic branch, those are the green fibers here, and you can see the sympathetic branch interacts with the SA node, the AV node, but also the myocardium, so the muscles of the ventricles, or the muscular wall of the ventricles. And so because the sympathetic branch is fight or flight, it is going to cause an increased depolarization rate of the SA node. It's actually going to speed up heart rate. So when you're exercising, when you are out there running, for example, you have to supply blood a little bit more quickly to the working musculature, so your heart rate has to speed up. So your heart rate's going to move from, let's say it's 60 at rest, and you go out for a light jog, maybe it's more like 100, right? Or 105, let's go with that. So anything above, I guess probably the easiest way to conceptualize this, particularly anything over 100 is going to be under the influence of the sympathetic branch of the autonomic nervous system. So sympathetic branch causes an increased rate of depolarization of the SA node, causes an increase in heart rate. It causes the ventricles to contract more strongly, and so that then increases the amount of blood that the ventricles eject with each beat, and so that is called stroke volume, the amount of blood ejected with each beat, so stroke volume goes up. Also causes reflexive relaxation in the coronary arteries, so it causes them to dilate or open up a little bit so that we can supply blood more readily to the myocardium, which is the heart muscle. In terms of chronotropic and inotropic effects, I just described those to you. So chronotropic effect is an increase in heart rate. Inotropic effect is an increase in contractility of the heart. So the sympathetic branch then speeds up heart rate and makes the heart contract more strongly. Both of those increase cardiac output, which is the amount of blood the heart pumps per minute. The parasympathetic branch, on the other hand, it's rest and digest. It's the brakes on heart rate. And so you can see the parasympathetic branch only has two connections to the SA node and the AV node. So the parasympathetic branch doesn't have any effect on contractility of the heart, but it does have an effect on heart rate. So the SA node taps the brakes, the parasympathetic branch taps the brakes on the SA node, causes it to depolarize fewer times per minute, and thus decreases your heart rate. So again, if we use 100 beats per minute or 100 depolarizations by the SA node as our starting point, anything lower than that is because you're tapping the brakes using the parasympathetic branch because you're resting and digesting. Anything higher than 100 is sympathetic influence, causing you to increase heart rate over 100. So if you go from resting, and let's say a heart rate of 60, to light exercise, so you're leaving class and moving fairly slowly, and your heart rate then increases to more like 80, basically what you've done is you've reduced the parasympathetic influence. You've stopped hitting the brakes as hard. And then if you need to start moving faster, then you'll remove the parasympathetic influence and activate sympathetic influence to increase your heart rate over 100. So that's how those two work together. Relevant terms that would be good for you to know: tachycardia is your heart rate's too high. And so that's over 100 beats per minute. And so usually those are used clinically like at rest. Somebody is tachycardic, meaning that just at rest they have 100 beats or higher than 100 beats per minute, and we may or may not know why. Obviously, if they're exercising, we do know why. And then bradycardia is less than 60 beats per minute, so that's a heart rate that is a little bit too low. Now, if you have somebody that's a well-conditioned athlete, it's not uncommon for somebody to have a resting heart rate in the 50s or even the 40s. The lowest resting heart rate that I'm aware of, supposedly, Lance Armstrong, at the peak of his Tour de France winning days, had a resting heart rate of 38. And again, so that would be due to lots and lots of conditioning, but also lots and lots of red blood cells in order to be able to deliver oxygen. So because of that, you wouldn't need quite as high of a heart rate. So anyway, getting off in the weeds a little bit. So just because somebody has a resting heart rate below 60 doesn't mean it's a problem. If they're a well-conditioned athlete, we would expect that. But if they're not and they have a resting heart rate of like 40, maybe that's problematic. All right, blood vessels. Arteries go away from the heart, veins come back to the heart. They are structurally different. So arteries are the high-pressure system. So when they're carrying blood away from the heart, they are carrying blood other than the pulmonary arteries. They're carrying blood that comes through the aorta out of the left ventricle. So the left ventricle contracts really, really strongly. And so arteries then have to be able to expand in response to that really strong contraction, that bolus of blood, if you will. So they have to be able to expand, and then they have to be able to recoil back to their original configuration to propel blood along the system. So the arteries are really elastic, and they have an extra layer of muscle around them that you can see here. And so that enables them to, or a really thick layer of muscle there around them, that enables them to recoil more effectively. What else about them do I need to emphasize? That's probably good for the NSCA. Capillaries are where gas exchange takes place. So here's our little capillary network down here. Capillaries are typically only one cell layer thick, so they're just the endothelium, which makes it really easy for oxygen to diffuse out of the capillary and into the underlying tissue. So that's where our nutrient and gas exchange take place. And then we work our way up from the capillaries to the venules and then eventually to the veins. The venules are not pictured on this diagram, but that's okay. So venules and veins are going to carry deoxygenated blood back to the heart. Again, other than the pulmonary circuit, they're going to carry deoxygenated blood back to the heart. They are a low pressure circuit, or they're low pressure vessels. So for example, if you are able to see the veins on the back side of your hand or on the posterior aspect of your hand, you can oftentimes just kind of mash down on them a little bit with one finger. You don't have to push very hard, but you can push down on them a little bit and you can kind of wring blood out toward your fingers. And then if you let go, the blood will return, right? So they're pretty obviously a low pressure system because you can kind of wring blood out of them. You obviously can't do that on the arterial side. But because they're a low pressure system, they also have valves in them to prevent backflow, particularly in the lower body. And so you can see the valve here. So what will happen is, let's say this vein is in our calf musculature. So the calf muscle will contract, push blood past this valve, and then it'll relax, which gravity would tend to pull it back down toward the foot. But because of this valve, that the backflow is limited there. And so that helps keep blood moving back toward the heart. Speaking of blood, so remember that blood has two primary components, plasma and red blood cells. The plasma is the fluid that the red blood cells float in. So if you are dehydrated, like after exercise, where a lot of those fluid losses have come from is a reduction in your plasma volume. So your blood has effectively gotten thicker, becomes harder for the heart to pump it, which is why you've heard that being dehydrated is not helpful for performance, because it becomes, your blood gets thicker and again, it becomes a harder workload on the heart. The red blood cells, remember that they are, so they float in the hemoglobin, not in the hemoglobin, man, all right. The red blood cells float in the plasma, there we go. And a protein inside of your red blood cells called hemoglobin, often abbreviated capital H, lowercase b. Hemoglobin actually binds to that oxygen and carries it inside of the red blood cells, and then releases it when we get to the capillaries, when we get to the tissues that need that oxygen. Hematocrit is the proportion of blood that is comprised of red blood cells. So for example, in men, average hematocrit is 47%. And what that means is that 47% of their blood is red blood cells. Give or take 5%. So it may be as high as 53 or as low as 42, but average is somewhere around 47% for men. And it's a little bit lower in women because women don't have as much testosterone. And one of the things that testosterone does is increases red blood cell production. And so for women, the average is a little bit lower at 42%, but again, plus or minus 5. So women on the high end may have 47% hematocrit, but on the low end may be more like 38%. So again, hematocrit, percentage of your blood that is comprised of red blood cells. And last, onto the respiratory system. Again, trying to make this not too long. The components of the respiratory system there, the primary thing to know about the respiratory system is that it is responsible for gas exchange. It's responsible for getting oxygen inside of the lungs and for moving oxygen from the lungs into the blood, and then going the other way, taking carbon dioxide from the blood moving it into the lungs, and then expiring it. So you can see the major organs here. There's a diagram of those. And the last thing to know, yeah, it's my last diagram. Last thing to know is that gas exchange takes place in the lung passively based on pressure gradients. So when we breathe air in, the pressure of oxygen inside of the alveolus is, so this PO2 is a partial pressure of oxygen. So that partial pressure is about 100 millimeters of mercury. The partial pressure of oxygen in the venous blood is only 40 millimeters of mercury. So oxygen is going to go from high pressure in the alveolus into low pressure in the venous blood that's returned until we reach equilibrium, till the pressure is the same. And then that is our fully oxygenated blood with 100 millimeters of mercury partial pressure of oxygen. Carbon dioxide goes the opposite way. Partial pressure of carbon dioxide, because that's a byproduct of the citric acid or the Krebs cycle. We produce carbon dioxide as a byproduct of breaking down carbohydrates, fat, and protein. And so we have to get rid of that carbon dioxide. And so we do that by breathing it out. So the partial pressure of carbon dioxide in the venous blood is higher than in the air that we breathe in, because again, it's a byproduct of energy production for us. So we're going to push carbon, or that pressure difference is going to push carbon dioxide from the venous blood into the alveolus, and then we're going to breathe it out. And that is gas exchange, at least as far as you have to know it for the NSCA. All right, so that's the end of chapter 1. So then hopefully you'll have read chapter 1, taken some notes on it, and watched this video. And then we're going to move on to chapter 2, or hopefully you'll have moved on to chapter 2 in a couple days, and then taken your first quiz by the end of the week. So good luck on the quiz, and I look forward to seeing you back for the chapter 2 talk.